Morning.
Morning. Good morning.
Kind of. It's not morning anymore. Never mind. Okay. First, any questions or concerns? We are into our last 2 weeks of class. And I'm realizing we are not going to be able to do chapter 15. I'm guessing most likely not going to be able to go through chapter 15 like we do in this face-to-face discussion format. If we have time, like next Thursday, we'll look at it some more. However, bear with me just for a second here. I posted 2 recordings here. And they are right here. It is not a really long chapter. Altogether, the recordings, I think, are about an hour and a half, maybe. One second here. They are right here. And there's 2 of them. The first one I think is about 30 minutes. The second one is about 50 minutes. I'm going to ask you to watch them on your own time. Today, I want to finish the special census and then plan on starting chapter 11 on Thursday. Any questions?
When do you think you'll post the final exam?
I will post it. Let me, I want to make sure that it's got what I want in it. And I don't have time to even look at it until this weekend.
Okay, that's fine.
But I will post it this weekend.
Okay.
I need to post the 3rd exam first. Let me post that and then I'll post the final. So you're going to have one more exam that's going to cover chapter 15, 16, and 11. And then the final is comprehensive. Any questions about that?
What do you mean by comprehensive?
Meaning it covers all of the chapters we've covered this semester.
Okay. So it's going to be the same as the exam, but it's like all the chapters?
Yes.
Okay.
Professor, please, when do you want us to do the 3rd exam?
Wait, say it one more time.
When do you want us to do the 3rd exam, exam 3?
The 3rd exam, as soon as I get, it'll get posted as soon as we finish chapter 11.
Okay.
And so it probably will not get posted until next week.
Okay.
Because we won't finish chapter 11 until next week.
And I have one question.
Oh,
I've done the chapter 16, the review, but I haven't seen the practice or the quiz available online.
Are they not available? Here they are right here.
No, the chapter 16.
Oh, the chapter 16.
Yeah.
I thought I posted them here. It doesn't mean I did. I thought I did. I will get them. Bear with me just for a second. Let me look. I thought I posted them. Eventually. They are not here. I will get them posted within the next hour or 2 hours.
Okay, thank you.
Professor, please, what
Yes.
of chapter 15? As it due?
Say it again.
Chapter 15 quiz and the practice.
They will be due if looking at the content section here. The practice is due December 8th.
All right, thank you.
The quiz is most likely going to be due that same day. I will make that, if it's not, it'll be due that same day, Toyin.
Okay, thank you.
You're welcome. Other questions? One second, let me look at the quiz. Oh, you're right, it doesn't even have a due date. I'm gonna make it due the December 8th. Don't know why I didn't include a due date there. It has a due date now, Toyin, December 8th.
All right, thank you.
You're welcome. Let's see here. I want to finish chapter 16 today. And like Kevin just asked me, I will get both the practices and the quizzes posted today. No. Sorry, give me 1 second here as I can't see any So the outer ear. So remember, there's three parts to the ear. There's the outer ear, the middle ear, and the inner ear. The outer ear and the middle ear are concerned with the transmission of sound vibrations. The inner ear is involved with converting those vibrations to nerve signals, meaning a series of action potentials. The outer ear includes the pinna, includes the pinna or the auricle, which is the outer ear here. The external auditory canal, which is made of from the external acoustic meatus, that bony marking on the side of your face. Within the auditory canal, there are ceruminous, and sebaceous glands with dead skin cells that produce cerumen. Cerumen is the anatomical scientific word for earwax. With the guard cells, it protects the outer end of the canal. And I can't remember if I talked about this or not the last time. So I grew up being hearing impaired, and I only got my first hearing aid when I was, how old was I? I was about 30 years old. So I've had it for just a little, I've had a hearing aid for just a little over 15 years. And and since I've had my hearing aid, I've learned a lot about earwax, because what I have is a hearing aid that's completely in the canal. So it fits completely in the canal of the ear. So it fits about right here. And one thing that these ceruminous and sebaceous glands do is they provide an immune function. If you have probably smelled it at some point, maybe. And if you haven't, count yourself as lucky. But earwax stinks. It's nasty. Once you smell it the first time, you'll never forget what it smells like. It's that nasty. Why does it stink so bad? The reason it stinks as bad as it does is it prevents things from taking up residence in your ear and growing. If you think about your ear, this ear canal that you have, it's a very warm, moist little area. And if you think about where would bugs and insects and parasites like to live, they'd probably like to live in the warm, moist area. And that is why you produce earwax, and why your ear produces earwax is to keep things out of the ear canal and to prevent things from wanting to be in your ear canal. And so now that I wear this little hearing aid in my ear, and let me go grab it really quick. I don't wear it when I'm teaching at home because I get easily distracted by the noises in my apartment. And so I don't wear it while I'm home teaching, but I wear it when I'm teaching everywhere else. But it's just about, altogether, it's just a little bigger than a dime. And it's probably one of the most expensive little devices I have outside of my car. This little hearing aid that I just got this last summer is $3,500, and it's not covered by insurance. But the only hearing aid that I can wear that works for my ears, and you can kind of hear it jingling there, but it fits completely in my ear canal. And I can't wear the kind that are over the ear. They don't, because of what my unique type of loss, which is a conductive loss, and maybe the fact that my ears are tiny. I can't wear an over the ear hearing aid. It mainly when I wear one, I can hear my hair really well, but that's about all I can hear that's better. Which I can tell you, it'll make you crazy after a little time listening to your hair all the time. But the reason I was telling you about this hearing aid is because when it's in my ear, like this one, my ear thinks that there's something in my ear, and it senses that there's something in my ear. And as an immune response to prevent something, whatever it is, from taking up residence in my ear produces more earwax as in response to the fact that there's something constantly in my ear. And if I get too much earwax in my ear, it can plug my hearing aid and possibly damage my hearing aid. So I'm constantly clearing out the earwax out of my ear to prevent damage to my little $3,500 device. But I've learned that's the reason your ear produces the wax, is because it's an immune response. It's protective response of your ear. So with the guard cells, these glands protect the outer ear and protect the outer end of the canal. The tympanic membrane, aka the eardrum, vibrates as sound waves enter the ear. Remember that sound is essentially a wavelength of air, vibrations of air. As that air vibrates and hits the eardrum starts vibrating to the same wavelength. In the middle ear, so now we've talked about the outer ear here. Now we're talking specifically about the middle ear. In the middle ear, or the tympanic cavity, there's 3 ossicles. These are small bones that amplify sound waves and transmit sounds to the inner ear. The first one has its handle, the handle of the bone, called the malleus, with the hammer, has its handle kind of attached to the tympanic membrane. And so when the tympanic membrane starts to vibrate, the malleus starts to vibrate too. The middle ear, the whole middle ear, and the tympanic membrane are innervated by the vagus and trigeminal nerves, and they're highly sensitive to pain. The malleus is attached to the incus. And the incus is attached to the stapes, which is attached to the cochlea via the oval window. There is a tube that exits the middle ear and drains into your nasal cavity, or not your nasal cavity, your nasal pharynx, which is the back of your throat, just posterior to your nasal cavity. It is called the eustachian, or the auditory tube. It allows for you to equalize. If you get a change in air pressure, it allows you to equalize atmospheric pressure. It allows you to equalize the pressure between the outer environment and that in your middle ear. If you've ever, when you fly, and you probably, even when you don't fly, if you change elevations, for example, in a car, you can often feel your ears kind of pop. What that is that your middle ear is equalizing the pressure to the outer environment. It's at an increased pressure, and so it needs to equalize, and that's what's occurring. If you've ever been on a plane, and you'll notice that parents who are used to traveling with children or learning to travel with children, small children or babies, what's one thing that they tend to do when the plane starts to arrive in at the airport, when the plane starts to descend?
They give them their pacifier.
They give them their pacifier, or they give them a bottle. And the reason they do that is to allow for them to start wanting to swallow and yawn, because it allows them to equalize the pressure in their middle ear. I can tell you, and people who struggle with ear, nose, and throat issues like myself, and I have a good friend who got the tip of her nose torn off by trampoline springs. I think I've talked about this, maybe I haven't, I can't remember. But she, when she was a lot younger, she got the tip of her nose torn off in a trampoline accident. And she will tell you it didn't hurt her at all. She says she didn't feel a thing. She just remembers there being a lot of blood. But since then, she cannot equalize the pressure in her middle ear when she changes altitudes. I also have the same problem, and I realize I don't have it to the extent that she does, because when I fly, I prophylactically take a pain medication, because I know that I'm not going to be able to equalize that pressure very easily. And the pain is terrible. It feels, I don't remember the pain of ear infections I had when I was younger, but I can imagine it being like that times about 10, because it's just a major pressure buildup in your middle ear. And I will get, before I started getting a little smarter and taking pain medication when I flew, I would get to the point of where I would be almost, curled over in my seat, wanting to claw my ear out because it hurt so bad. And so I started getting a little smarter and taking pain medication before I got on the plane. However, I learned talking with this friend that I can't complain, because one thing that I've never had happen when I've flown is I've never had my eardrum rupture because of the pressure. And this friend that got the tip of her nose torn off, her eardrum will actually rupture and bleed when she flies because the pressure is so bad. So the whole function of this tympanic eustachian auditory tube is to equalize that pressure. And one thing that you'll notice here, here's a baby, here's an adult. As within the baby, your face as you grow up starts to elongate, and the auditory tube becomes more vertical as your face elongates. It's more horizontal here, more vertical here. Why do babies struggle with more ear infections? Because of the inability for this to drain, fluid to drain from your middle ear into your nasal pharynx. So middle ear infections are a buildup of fluid in the middle ear. They're common in children because the auditory tube is short and it's horizontal. These infections can easily spread from the throat to the tympanic cavity and mastoid air cells. Symptoms are that fluid accumulates in that tympanic cavity, causing pressure, pain, and impaired hearing. They can spread, leading to meningitis. Chronic ear infections can cause fusion of the ear ossicles, that malleolus, incus, and stapes. And as a result, you can deal with hearing loss. This is my issue, at least in one of my ears, is that I have fusion of those ossicles. A tympanostomy, it's actually not that, is tympanostomy, is when they put tubes in the ears. What they do is they lance the tympanic membrane and insert a small tube, which allows for fluid to drain out of the tympanic cavity. If you look here, here's the eardrum. It actually I've seen what they look like. If you look up some pictures on Google, you can see them. They look just like this. They're very smooth. And a lot of times you can see the handle of the malleus attached to the eardrum. Because I told you that I dealt with chronic ear infections as a child. And I also had tubes put in my ears multiple times because of the ear infections. And I don't know how many sets of tubes I had. I think at least 5 sets. And when I was about 13 or 14, they decided that I needed to be done wearing tubes. First off, I was getting too old to be wearing tubes all the time. And they decided I just needed to be done wearing them. And so after the very last set of tubes, they just waited for the hole that was created by the insertion of the tube to close over, and it didn't. And so over a short period of time, maybe a long period of time, I can't remember, I was only 12 or 13 at the time. But they watched as this hole, rather than the tube falling out and the hole closing over, the tubes fall out over time, the hole stays open, and then normally it closes over. Well, mine, rather than closing over, actually started growing out. And if it had grown completely out, I would have become completely deaf in that ear. So rather, because it was growing out and not growing over, they went in and grafted me a new eardrum. And because every time you get new hearing aids, you get new ear exams. One of the first person that I ever saw to get a hearing aid let me, because he had a camera that was hooked up to a screen that I could see, let me see what that graft looked like in my ear. And I can tell you it's really funky. It's not smooth like this. It looks like I've obviously had a lot of work done on my eardrum. But when they went in and did that graft, that's when they saw that the 3 little bones were partially fused in that ear. Whether they're partially fused in the other ear, we don't know. But I only wear a hearing aid in my right ear because my loss is more significant in that ear. Whether the graft has to do with the fact that maybe the eardrum doesn't vibrate quite right is a case also. Who knows? Any questions? Within the inner ear, it is structured like a tube within a tube. There's a bony labyrinth which contains passageways in the temporal bone. There's a membranous labyrinth within that bony labyrinth, which is a flex sheet tube. The membranous labyrinth is filled with endolymph, which is similar to intracellular fluid in its contents and texture. This membranous labyrinth filled with endolymph is floating in perilymph, which is similar to cerebrospinal fluid in its texture. So if you look here, here's this tube within a tube. And the tube itself, the inner tube, just like if you're riding a bike, you have the inner and the outer tube. The inner tube is filled with endolymph, and the outer tube is filled with perilymph. And the same thing within the semicircular canals. There's the membranous labyrinth that's within the bony labyrinth. Another depiction of it. The cochlea meaning a rounded shell-like structure, is the organ of hearing. It coils around a screw-like axis of spongy bone called the modiolus. or modiolus. The vestibule, right here, the vestibule and the semicircular canals are the organs of equilibrium that are involved in your sense of balance. So looking more specifically at the cochlea, and then we'll focus on the semicircular canals. But right now, we'll focus on the cochlea. The scala media, the cochlear duct, is a middle chamber filled with endolymph. So this is the inner tube, is the scala media within the cochlea. It is called the cochlear duct, is the other name for it. It's the middle chamber that's filled with endolymph. It's separated from the scala vestibuli and the scala tympani by the vestibular membrane and the basilar membrane. Here's the basilar membrane. Think basal meaning basement, meaning the bottom membrane, and the vestibular membrane up here. And within this cochlear duct is the organ of Corti. If you think about a piano organ, it's similar in a lot of ways. It's the acoustic organ which is going to convert the vibrations of the air that are then converted to vibrations of the endolymph, which is then converted to nerve impulses by this organ of Corti. So this organ of Corti, or spiral organ, has epithelium composed of hair cells and supporting cells. The hair cells have long, stiff microvilli called stereocilia on them, on the apical surface. And they are resting and slightly embedded in a tectorial membrane that's gelatinous in texture, kind of like a gel, kind of like Jell-O, but a much stiffer form of Jell-O. Membrane made of like a gel-like substance that's kind of stiff. So here's the organ of Corti. Here's the hair cells. We have 3 rows of outer hair cells and 1 row of inner hair cells. These hair cells with their stereocilia are embedded within the tectorial membrane. The whole organ sits on the basilar membrane. So 4 rows of hair cells, 3 on the outside, 1 on the inside. The outer hair cells are composed of 3 rows of about 20,000 cells. And the function of these outer hair cells are to adjust the response of the cochlea to different frequencies. This is like cochlear tuning, or tuning a piano. Think of it like tuning a piano. They come in and they kind of tighten some strings and adjust other strings. If you play the guitar, you've done this before, violins, kind of tighten some things and loosen other things. That's the function of these outer hair cells. They increase the precision of sound sensation. Inner hair cells are a row of, there's only a single row of about 3,500 cells. And they're what actually provide for the hearing. If you look, the outer hair cells are a little elevated, a little over the inner hair cells, which allows them to allow for more precision of what's sensed by the inner hair cells. So here's the outer hair cells, here's the inner hair cells. Here's the tectorial membrane. Here's the basilar membrane. Here's the endolymph up here. And the tectorial membrane. The endolymph is a high potassium medium. Cochlear nerve is attached to that inner hair cell because remember, that's what's actually going to provide the hearing. The outer hair cells are going to tune the cochlea so that only some hearing sounds are sensed and not others, allowing for more precision in hearing. So thinking about this transmission of sound and its ability to generate action potentials in the hair cells. The tympanic membrane has about 18 times surface area of the oval window, which is where the stapes is attached to the cochlea. Remember, the stapes is one of the little bony ossicles. So the ossicle's function is to concentrate the energy of the vibrating tympanic membrane that's vibrating in rhythm with the air vibrations onto the oval window. That's the function of the ossicles. The ossicles and muscles that are attached to the ossicles also have a protective function called the tympanic reflex. This tympanic reflex is under the control of the trigeminal nerve, cranial nerve number 5. This, and remember, all of your reflexes are a protective response of your body. When a noise gets too loud, these muscles will pull on the ossicles, reducing the vibration. Therefore lessening the transfer of energy into the inner ear. Questions? This vibration of ossicles, the vibrations are going to cause pressure waves in the inner ear fluid of the perilymph. Which results in movement of the basilar membrane. So here's the basilar membrane right here. It starts moving up and down to the same vibrations as the sound vibrations coming in from the middle ear. And when it does that, it's going to start bending the stereocilia up against the tectorial membrane. These are mechanoreceptors. They open in response to being bumped. Mechanoreceptors. The stereocilia of the outer hair cells are bathed in this high potassium fluid. And because the potassium concentration is much higher outside of the hair cell, there's an electrochemical gradient produced, where outside of the hair cell is plus 80 millivolts. Inside the hair cell is a near negative 40 millivolts. And the stereocilia tips are embedded in the tectorial membrane. The stereocilium on the inner hair cells are mechanically gated channels where the tip link, remember there's 3 rows of stereocilia of hair cells, 3 rows of hair cells, where the tip link to the highest row, tallest row, is connected to the ion channels of the sidewalls of the other rows. The tip link connects one to the next one. So when the top one bends, the others bend too. If this one gets bent, then these get bent too. And remember that the tectorial membrane is right here. So as this basilar membrane starts vibrating with the air wavelength, ba-bump, 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 it starts pushing these tips up against the membrane that's lying up here. Opening these channels. What happens when you open an ion channel? You either depolarize or repolarize a membrane. And in this case, you're depolarizing the membrane. So as the tallest stereocilium is bent towards the tectorial membrane as the basilar membrane is vibrating, it pulls on the tip links and opens ion channels. As the potassium flows in, the depolarization causes a release of neurotransmitter and stimulates the sensory dendrites, generating an action potential in the cochlear nerve. Variations in the loudness cause variations in the intensity of the vibrations. A soft sound produces very small vibrations of the tectorial membrane. A louder sound makes the basilar membrane vibrate a lot more vigorously. So a soft sound is going to trigger lower frequencies of action potentials. A louder sound is going to trigger a frequency of higher action potentials, higher frequency. And the higher the frequency, the louder the brain interprets that sound to be. The pitch of the sound depends on which part of the basilar membrane vibrates. Remember that this membrane is this curled up shell. Right here, it's stretched out, but it's actually curled within the cochlea in actuality. At the proximal end near the oval window, where the membrane is attached to the middle ear via the oval window, that basilar membrane is more narrow and stiff. When that part of the basilar membrane vibrates, the signal is interpreted as being a higher pitched sound. At the distal end, or the farthest end away from the middle oval window of the basilar membrane, when that end of the basilar membrane vibrates, the brain interprets those signals as being lower pitched. Questions? Well, once the action potentials are initiated, where do they go? This sensory pathway for auditory signals involves 4 different orders of neurons. The first order neurons are the axons that are going to run from the cochlea to the cochlea nuclei in the medulla oblongata of the brainstem. The second order axons are going to travel from the cochlea nuclei of the medulla oblongata to the superior olivary nucleus in the pons or the inferior colliculi. Remember that corpora quadrumina in chapter 14? That's the inferior colliculi. The third order neurons are going to travel from the superior olivary nucleus or the inferior colliculi to the thalamus. Remember that the thalamus is that sensory gateway to the cerebral cortex. So fourth order neurons are going to travel from the thalamus to the primary auditory cortex. Remember, that's found in the temporal lobe. So the first order neurons travel from each of the cochlea to the cochlear nuclei on both sides of the medulla oblongata. Second order neurons can take one of 2 pathways. They can ascend from the cochlear nuclei to the inferior colliculi, which are involved in locating sound, detecting fluctuations in pitch, and auditory reflexes. Startle reflex. When you move, you turn your head towards a loud sound, or you jump. Or they can ascend towards the superior olivary nucleus and be involved in binaural hearing, which is the way you localize sound. The way that you localize the sound is how long does it take for the sound to reach this eardrum or this eardrum? And your brain can detect the very tiny difference in time it takes for one eardrum to detect the sound and the other eardrum to detect the sound. That's the way you localize where sound is coming from. I can tell you that being hearing impaired, a lot of times in a classroom, I will struggle to hear which of my students is talking, because I struggle to localize sound. Because of the differences in the two eardrums. The superior olivary nucleus is also involved in that tympanic reflex, protecting the ear ossicles from overly vibrating. Third order neurons begin in the inferior colliculi and synapse on the fourth order neurons in the thalamus. And the fourth order neurons are going to travel to the primary auditory cortex of the superior margin of the temporal lobe, allowing for a conscious perception of sound. Any questions? Conductive deafness, hearing loss. Conductive deafness is conditions that interfere with the transmission of the vibrations to the inner ear. So this is issues with the outer ear and the middle ear. Can be the result of a damaged tympanic membrane, like a rupturing of the membrane. I told you about why I had to get a tympanic graft, was because of damaged tympanic membrane. Otis media ear infections can cause, chronic ear infections can cause conductive deafness because of that otosclerosis. Blockage of the auditory canal. Or otosclerosis. Sensorineural deafness, which is usually the deafness associated with age, is death of the hair cells or any nervous elements concerned with hearing. This is more specific to the inner ear. I want to watch a video really quickly. On the ear. Here, or is it? Yep, it is here. I have to remember which one it is. Do I not have it here? All right, I'm going to go to another class. Bear with me here just for another class site because it's not there. And I'll transfer this link over.
Writing's not that easy, but Grammarly can help. To hear, our ears must capture sound, transmit it to the organ of Corti, and translate it into neural impulses to be delivered to the brain. The organ of Corti is located in the cochlea, a spiral, three-chambered, snail-like structure, 10 millimeters wide, within a bony matrix. The organ of Corti extends from the anterior part of the vestibule and coils for about 2.5 turns around a bony pillar called the modiolus. In cross-section, the uppermost chamber is called the scala vestibuli. The oval window is situated at the base of this chamber. The lowermost chamber is called the scala tympani. At the base of this chamber is where the round window is located. Both the scala vestibuli and scala tympani contain perilymph. Between the scala vestibuli and scala tympani is the scala media. This houses the organ of Corti, which is referred to as the receptor organ of hearing. The scala media is filled with endolymph. The scala media includes structures from the tectorial membrane, basilar membrane, and hair cells, which sense the mechanical forces. The hair cells are located between the tectorial and basilar membranes. Approximately 16,000 hair cells are within the cochlea. There are two kinds of hair cells, inner and outer hair cells. 95% of the afferent fibers are from the inner hair cells, the sensory receptors that communicate with neurons from cranial nerve 8. Outer hair cells receive mostly efferent input from the superior olivary complex. The filamentous structures that connect the tips of adjacent stereocilia are known as tip links. These are thought to amplify the forces in the area of the molecular sensors. How does sound enter the cochlea? Compression hits the tympanic membrane, causing the stapes to transfer force to the oval window. The sound travels down the scala vestibuli, around the helicotrema, to the scala tympani, allowing its fluid perilymph to mix. From there, sound moves to the round window. High frequencies are encoded at the base, and low frequencies at the apex. It is this property that leads to the tonotopic map along the basilar membrane. The manner in which the basilar membrane vibrates in response to sound is the key to understanding cochlear function. The hair cells are located between the tectorial and basilar membranes and are stimulated by the shearing force between the two caused by the pivot point of the two membranes. The pivot point of the basilar membrane becomes displaced. The tectorial membrane moves across the tops of the hair cells, causing the stereocilia to bend. The ionic environment of the compartments plays a critical role in signal transduction. The apical portion of the hair cell is bathed in high potassium solution, and the base of the hair cell is bathed in potassium pore solution. This causes the opening of mechanosensitive channels, allowing potassium to flow into the cell, leading to depolarization. This in turn opens calcium channels at the basal end of the cell, leading to vesicular transmitter release to stimulate the nerve.
So that,
Because the relative voltage and potassium levels are low at the base of the hair cell,
that is when you remember this calcium release, that's the neurotransmitter release. You remember that calcium is really involved in exocytosis of neurotransmitter. That's it right there.
because the relative voltage and potassium levels are low at the base of the hair cell, potassium flows out of the cell. This establishes the potassium flow through the cell is used for both depolarization, potassium in at the apex, and repolarization, potassium out at the base of the hair cell.
So any questions? We want to get started on balance before the end of the class here. Oh, wrong one. So the other major sense that your ears are involved in is balance. We have ciliated equilibrium sensory receptors that are located in what are called the saccule and the utricle, which sense tilting of your head one way, other way, other way, other way. Semicircular canals that sense rotation when you're spinning. There's two types of equilibrium, or three types. Static and dynamic equilibrium and linear acceleration are going to be detected within the vestibule, which contains the utricle and the saccule. Within this area, there are receptors and otolith fluid. Otolith means like a crystallized substance. Oto means rocks. Otolith means crystals. Semicircular canals also have these little otoliths in them, but they are involved in sensing dynamic equilibrium and angular acceleration. Linear acceleration, you're in your car and all of a sudden you hit the gas and you start going, you go from 30 to 45 to 60 miles per hour. That's sensed within the vestibule. When you spin a little tiny kid. We've all done this at some point or seen it. You grab a little kid by the arms and you say, let me spin you. And you start to spin them by their arms. And then you set them on the floor and say, walk straight. And have you ever noticed how they can't do it? They can't walk straight once you do that. That's because of the semicircular canals. Static equilibrium within the vestibule. When the head is tilted, the otolithic membrane, so this is a membrane that contains a lot of rocky crystals in it. Then the stereocilia, just like we saw in the ears, we have hair cells here that have stereocilia, allowing for depolarization of those receptors. So the reason you can sense your head tilting is because of a depolarization of hair cells via this otolithic membrane sagging on those hair cells. So this otolithic membrane is similar in structure and context to the tectorial membrane in the ear, in the cochlea. Linear acceleration is when, rather than the otolithic membrane sagging forward, it's lagging behind, stimulating the hair cells. The saculi is nearly vertical and responds to vertical acceleration. When you go from a go really high up in the air and go really down really fast. Ever been on those kind of rides that drop you? Those rides at fairs where they drop you from a big height? That's what's being sensed in the saculi. Questions? We will definitely finish chapter 16 on Thursday and start chapter 11. Have a wonderful 2 days.
And you too. Thank you. Bye-bye.
Thank you.
Bye.
Thank you.
Welcome.